Good morning. My name is Kirby Stafford. I am the Chief Entomologist and State Entomologist at the Connecticut Agricultural Experiment Station in New Haven, Connecticut. Welcome to the Dutchess County Tick Pest Force Education webinar. I'm going to start off this morning talking about biorational and ecological strategies for tick control and the prevention of tick-borne disease. And this is a review of a lot of the research that's been done over the years to try to manage the risk of exposure to ticks. And I'm going to start this presentation with a quote from the book Biology of Ticks by Dan Sonnenschein. Few agricultural or health problems confronting human societies have proved as intractable as control of ticks and the many diseases they transmit. Tick control has been and is heavily dependent upon chemical toxicants termed acaricides. And to give a little bit of a historical perspective before we get into some of these strategies, in 1893, Theobald Smith and Fred Kilbourne proved that Texas cattle fever was caused by a protozoan, Babesia bignimina, and it was transmitted by a one-host tick, Boophilus annulatus. And this led to a tick eradication campaign, which largely eliminated this tick from the U.S. by 1960. But these findings stimulated the work on Rocky Mountain spotted fever, which was the first human tick-borne disease identified in America. It was taking a terrible toll on settlers in the Bitterroot Valley of Montana in the late 1880s, which led to the establishment of the Rocky Mountain Laboratory in Montana in the early 1900s. Now, DDT was used for large-scale control of the tick Ixodes persilicatus, which actually transmits Lyme disease in Russia. And this was done in the 1950s to reduce the incidence of tick-borne encephalitis. But really, it was the recognition of Lyme disease in Lyme and Lyme, Connecticut in 1975 that really re-triggered a focus on human tick-borne diseases and tick control, even though it had a long history in Europe. And it was Dr. Willy Bergdorfer, who ironically was at the Rocky Mountain Laboratory, established because of Rocky Mountain spotted fever, who identified the spirochete Borrelia burgdorferi in the black-legged tick Ixodes scapularis, and that was done in 1982. When serological tests became available, the Connecticut Agricultural Experiment Station, working with the Connecticut Department of Public Health, offered free testing to physicians throughout the state of Connecticut, and it gave us our first glimpse of the distribution of Lyme disease in 1984 and 85. As you can see, at that time, most of the cases were still in southeastern Connecticut, east of the Connecticut River. But of course, since then, in the 30 years since that study was done, we've seen the tick and Lyme disease expand geographically quite dramatically throughout the northeastern, mid-Atlantic, and upper Midwestern states, as well as cases occurring elsewhere in the U.S. And along with that, we've seen a dramatic and steady increase in the number of reported cases of Lyme disease to the Centers for Disease Control and Prevention, running around, like last year was around 36,000 cases, but the true incidences are probably around 300,000 human cases each year. But Lyme disease isn't the only disease that we're dealing with. There are at least 13 other tick-associated human diseases in the U.S., 5 of which are associated with our black-legged tick Ixodes scapularis. So we do have an increasing issue with not just Lyme disease, but a host of other tick-borne diseases. So when we talk about tick control, in the absence of a Lyme disease vaccine, we basically have several approaches, and they're broadly classified as our ecological approaches, which are cultural, like vegetation modification to reduce the suitability of the habitat for ticks and its hosts, host manipulation, and then we have a variety of pest control agents. These can be conventional or synthetic chemical pesticides, application of pesticides to hosts, and also a lot of renewed interest in biologically or biologically derived pest control agents, which include what we call our minimum risk 25B botanical-based pesticides, host-targeted vaccines, and microbial control agents. And I'll be discussing some of these as we go through the presentation, and there'll be other speakers later that will also be addressing some of these as well. Or another way to look at it, to summarize some of these strategies as I go through these, of course, education and personal protection measures are important, and I'm not going to be discussing those in this presentation. But the approaches have included landscape modifications, chemical control, whether it's synthetic acaricides, biopesticides, or the so-called natural or botanical compounds, host reduction or exclusion, host-targeted acaricides, and host-targeted vaccines. And a lot of the efforts have focused in the residential community. You think about where is the risk of exposure to ticks and where are most people actually acquiring Lyme disease and other tick-borne diseases. And the information that's gone out, we've thought of homes and basically in terms of where the ticks are. They're mainly found in woodland areas. That's your tick zone. Density of ticks is much less in more open portions of the lawn around homes. So the idea is to using landscape or spray barriers to reduce the likelihood of encountering ticks, because about 3/4 of the ticks are acquired at home. And surveys that were done by Connecticut Stafford Health Department and associated with ticks that were being submitted to the experiment station for testing, 74% said they picked up the ticks right outdoors at home. But a good substantial still were picking them up away from home. That was 21%. But if you look at the activities, 47% of those ticks were associated with play activities outdoors, and 18% with yard work, and 12% with gardening. And interesting, this kind of corresponds with the age distribution of Lyme disease cases in the United States, where the highest incidence is in children, lowest incidence is in the 20s, and then you'll see a steady increase in cases in people in their 30s, 40s, and 50s, which probably corresponds with home ownership and those gardening and yard work activities. So there's basically where your risk. So a lot of the research is focused on controlling ticks in that environment. But interestingly enough, higher tick counts have also been associated with exotic invasive forest understory than with our native understory. So the occurrence of things like barberry and other invasive plants in our forests or in the woodlands around our home has actually enhanced the habitat for ticks and the rodents that carry them. And if you'll notice in this slide, from the study that was done in Maine, the densities of adult ticks in blue and nymphal ticks in the reddish color there under the first column, exotics, is practically to almost twice as high as those that were in native shrubs, and substantially higher in areas that had no vegetation at all. And here in Connecticut, it was found with Japanese barberry, which has formed dense stands in part of our forests and actually prevented a lot of forest regeneration. Questing adult black-legged ticks abundance was greatest in areas where you had dense Japanese barberry stands. And the number of larval ticks on the mice was much greater in areas with barberry. And when barberry was reduced from 62% of the forest cover to only 3% by mechanical cutting and burning in controlled areas at 3 sites in Connecticut, it reduced the density of the Lyme spirochete-infected adult ticks by nearly 60%. And the number of ticks in areas that had no barberry were even lower still. So if you have a lot of barberry right in your home, in the woods behind your home, that is definitely a high-risk tick habitat. But spraying continues to be the primary method of controlling ticks in most residences. And most professional applicators will use one of the synthetic pyrethroids, such as cyfluthrine, which is known as Tempo, or bifenthrin, which is Talstar. But there's been a lot of interest in alternatives, particularly in biopesticides. And these are certain types of pesticides derived from natural materials such as animals, plants, bacteria, and certain minerals. And the EPA has 3 major classes of biopesticides, 2 of which are relevant to us. These are microbial pesticides, which consist of a microorganism such as a bacterium, fungus, virus, or protozoan as the active ingredient, or biochemical pesticides, which are naturally occurring substances that control pests by non-toxic mechanisms, and this includes plant abstracts. That's how the EPA classifies it. And if you look at the number of compounds registered with the EPA, you'll see that most of the registrants, 89 of the compounds, are conventional pesticides. There's 1 microbial registered with the EPA and 6 biochemically based pesticides registered with the EPA. Of course, the microbial agent is the fungus Metarhizium anisophili, the strain 52. This is a naturally occurring soil fungus. It's an entomopathogenic fungus that attacks a number of different kinds of insects. It is being manufactured under the trade name at the moment, MET52 Bioinsecticide from Novozymes Biologicals, Inc., in conjunction now with Monsanto. And it was actually initially developed here by a small company in Connecticut. We did trials with it in 2002, and then when Novozymes picked it up, we resumed those trials in 2007, and we got 53 to 73% control in our original trials. This led to full EPA registration. It's registered in all states, and the company is planning a limited commercial launch this year. I don't know exactly where or who it will be available to, but it will start becoming available. And there's now a non-refrigerated formulation, which should make it much more easier to use. Now, some pesticides have been deregulated by the EPA. These are what we call the Federal Insecticide, Fungicide, and Rodenticide Act 25B minimum risk compounds. They're exempt from federal registration, but they're not exempt from state registration. And some of the examples of these include rosemary oil, peppermint oil, thyme oil, cedar oil, garlic oil, and geranol. And there are other compounds like cinnamon and other things that are on the list as well. But the number of these compounds have been formulated into products for mosquito control, tick control, deer repellents, things of that nature. But some of the issues with these botanical-based products is that, you know, when you're talking about a particular oil, the compound can be quite variable and of unknown composition because the actual makeup of those essential oils will depend upon the source, like the plant species, method of extraction, and things like that. There's questions concerning the efficacy of the actual oil versus what the proportion of specific components in that oil are that may or may not be active. A lot of them are volatile, and there's a lack of persistence. Some of these oils do have some phytotoxicity issues, so you have some risk of possibly burning the vegetation with some of the compounds. And at this point, and there is research being done, there's little independent data on the efficacy or effects of non-targets of these compounds. So they are pesticides. They kill ticks. They will probably impact some other things as well. Some of the studies that have been done, we looked at a product called Mosquito Barrier, which is 99% garlic juice. In our trials, we found it suppressed or controlled tick activity by 37 to 59% for 1 to 3 weeks. Up in Maine, a study was done looking at eco-exempt IC2, which is no longer available, but it consisted primarily of rosemary oil and peppermint oil, and it provided very good control of nymphal and adult Ixodiscapularis ticks for about 3 to 4 weeks. And then from 2008 to 2010, studies were done with Nucatone. This is an essential oil from Alaska yellow cedar. There's a synthetic version, and it can also be extracted from grapefruit. It's highly repellent and toxic to Ixodiscapularis. It's a food-grade flavor additive, and it's used in the soap and cosmetic industry. So it has a very good safety profile. And in the field trials in Connecticut and New Jersey, it provided 80 to 100% control depending on the formulation and the spray pressure used, so forth. But the problem is it's used in minute amounts, and it is very expensive, it's very volatile, and it is slightly phytotoxic. So there are some issues that will be addressed, primarily having an economic source of this material to see it developed as an actual acaricide. Of course, another approach that has been taken is host-targeted tick control, focusing either on white-tailed deer, which is the main reproductive host for our black-legged tick, or on white-footed mice, and to a less extent eastern chipmunks, which are primary hosts for the subadult stages of the tick as well as reservoirs for the Lyme disease spirochete. The approaches with deer have included exclusion, reduction, and treatment with topical acaricides through a device called the four-poster. And for the mice, the focus has been on the topical application of acaricides to the mice through, most recently, a bait box, and there'll be more about that later in the program as well. But when you look at approaches taken with either deer or mice, one of the aspects is the size of their home ranges. Anything targeting mice, which have a home range of only about roughly a half acre, maybe an acre at the most, obviously the intervention has to be applied multiple times, even around a single residence. With deer, they have a urban home range running up into potentially several hundred acres. So you're impacting a much larger neighborhood scale with those kinds of approaches. So with deer, some of the first studies that were done were looking at deer exclusion. I did a study in Lyme and O-Lyme where we had a couple of properties that were surrounded by a 7-strand high-tensile electric deer fence. And without deer coming in, there was no larval ticks, so we had a 100% reduction there, an 84% reduction in nymphs, and a 74% reduction in adult ticks. And another study done down in New York by Tom Daniels and Rich Falco and Associates found very similar results as well. In terms of the topical application to deer, there was a Northeast area tick control project that was conducted in 5 states, Rhode Island, Connecticut, New Jersey, New York, and Maryland. It was a 7-year project using this 4-poster device, which consisted of a bin of corn. As the deer come in to feed, they would brush their head up against these paint rollers that were treated with a topical acaricide. And at the end of the study, a meta-analysis of the entire project found that there was a 71% control of the nymphal black-legged ticks by the 6th year of the project, and the entomological risk, meaning the infected ticks, was reduced by 68%. And taking a quick closer look at that, you can see that the percentage control increased steadily with each year of treatment. And by the 6th year, we achieved that 71% overall control. But 2 years after treatment of the deer was stopped, you can see that the percent control started to drop quite dramatically. Deer reduction is another approach that has been examined in a number of studies. The latest study that's been published was one that was done here in Connecticut in the community of Montford Cove in Groton, Connecticut. This is a private residential community within the town of Groton. And controlled deer hunts were implemented in that community in November of 2000, and you can see that the deer density shown by the dotted line steadily decreased. And with a 2-year lag, given the 2-year life cycle of tick, you can see that nymphal abundance also dropped as deer density declined, and this relationship was highly significant. If you look at the number of reported cases of Lyme disease in the Montford Cove community, you can see with deer density plotted again is the dotted line in this graph. And the number of cases of Lyme disease within that community is the solid line. And you can see that the number of cases there declined with the drop in deer density and number of ticks within that community. To summarize that particular study, deer were reduced from a density of about 122 deer per square mile to 13.2 deer per square mile. That was an 87% reduction in the population. Deer sightings by the residents significantly declined. There was an overall 76% reduction in tick abundance. But interestingly, there was an 80% reduction in resident-reported cases of Lyme disease from before until after the hunt was initiated. So the conclusion of this study that reducing deer to a density of around 13 deer per square mile can significantly impact the number of infected ticks in the environment and the human risk of contracting Lyme disease. And more dramatically still, on Muskegon Island off of Maine, the deer were actually eliminated. This is a 237-hectare island off the coast of Maine. Deer were actually introduced to that island in 1955. They're not native there. Deer density was at least 100 deer per square mile. And in 1 year, in 1996, 13% of the residents contracted Lyme disease. So the community made a decision to have all the deer removed, which was carried out between 1996 and 1999. And subsequent to that, no subadult Ixodiscapularis ticks have been found on Norway rats, and even fewer adults have been flagged. And interestingly, there are no mice on this island. The rats served as the host for the larval and nymphal ticks, as well as the reservoir for the Lyme spirochete. And since then, the subsequent studies have shown or suggested that all the adult ticks that they found since then appear to be due to bird-imported nymphs, because looking at the birds, they are carrying ticks onto the island. So the elimination isn't total because of the introduction of ticks by birds, to the island. So one of the real dilemmas as we look at tick interventions is, can we reduce ticks? Yes, but documentation of having an actual impact on disease has been much more difficult. There's a number of factors to that, obviously with reporting issues, tracking cases, things like that. But it is the couple of deer intervention studies have documented a reduction in human Lyme disease. And I've already alluded to the study in Mumford Cove where we saw an 80% reduction in resident reported cases of Lyme disease from before until after the hunt was initiated. And then in that 4-poster study in Old Lyme, Connecticut, which was the community here in Connecticut where the study was conducted, a subsequent analysis of the data, we found that in the original treatment area with the control town, Lyme disease reduction was significant. So by treating the deer, we did see an impact on disease as well. So when you look at the overall studies that have looked at deer reduction, most of these studies have all been done on islands or geographically isolated tracts, simply because it's much more easier to try to get a handle on the deer population in those types of communities. There's Great Island study that was done a long time ago, not much follow-up information on that, but it did reduce ticks. Crane Beach, there was a reduction study from 44 to 11. There was a reduction in the number of ticks on mice. Monhegan Island obviously had a dramatic impact on the number of ticks on the island, had an impact on Lyme disease. Supposedly there's only been one human case of Lyme disease on the island since then, but the numbers are so low it's really hard to show any statistical impact. Bluff Point State Park in Connecticut and Bridgeport, both of those don't really have a human element that can be documented, but nonetheless, there was a dramatic tick reduction in the number of ticks there. I've already talked about Mumford Cove. And in Bernard Township, New Jersey, there was a study where they reduced deer from about 118 deer per square mile to 63 deer per square mile. Did this impact the number of ticks? No. 63 deer per square mile is more than enough to sustain a high tick population. And of course, and it's unknown whether it had any impact on disease. And one of the issues when people talk about managing deer as an approach to control Lyme disease, it's not an easy thing to do. In a review that was done by Dr. Scott Williams, our wildlife biologist here at the experiment station, he looked at 3 controlled hunting programs that were carried out in New Jersey and Pennsylvania. And these areas had initial deer populations of 78 to an astonishing 207 deer per square mile. And after 3 to 10 years, hunting resulted only in a decrease to around 44 to 46 deer per square mile. So they has concluded that existing regulated hunting techniques were insufficient to maintain deer densities below that 30 to 40 deer per square mile range, and other measures would have to be taken if you want to get it down below that. And as you saw in Mumford Cove, we saw an impact when we got the deer down to only 13 deer per square mile. In another approach that's taken targeting mice, we have the fipronil bait boxes. And you'll hear more about these a little later in the program. These boxes contain a non-toxic food block and a wick that contains fipronil. Fipronil is the active ingredient in the product Frontline for flea and tick control for dogs and cats. And one application to a white-footed mouse entering one of these bait boxes will result in about 30 days of complete tick control on that mouse. So in other words, instead of the mouse picking up ticks and infecting those ticks, it'll pick up any ticks in the environment and actually end up killing them. In the initial study that was done on Mason's Island in Mystic, Connecticut, we saw quite dramatic reductions in the number of nymphs that were collected at the treated sites. In the top graph, you can see the untreated areas. In the red, there's the number of ticks in the areas that were treated in 2000, 2001. And in the purple, you can see the areas that of the island that were initially treated in 1999. Very dramatic drop in the number of ticks. And essentially, by eliminating a lot of ticks, there was also a dramatic decline in the infection, Borrelia infection in the host Paramiscus, Leucopus, white-footed mice on the island as well. And although not published, subsequent follow-up to this 3-year assessment, ticks were essentially eliminated from the island by treating all of the residents on the island. So it was quite successful. And the real key there is probably coverage. Obviously, the more of the animals that you reach, the greater the impact that these boxes are going to have. Now, another approach that is being taken, there was a study that was published last year looking at a reservoir-targeted vaccine. And again, there'll be more about this later in the program. An E. coli expressing Ostfeld vaccine was deployed as an oatmeal bait over a period of 3 to 5 years at 4 plots of land in New York. After 2 years of feeding the mice the vaccine-laden oatmeal, there were 23% fewer nymphal Ixodes infected with the Lyme spirochete, Borrelia burgdorferi. And after 5 years of treatment, there were 76 fewer nymphs that were infected. So this would suggest that the application of a reservoir mouse-targeted vaccine could reduce the prevalence of infection in our reservoir hosts and also subsequently in the ticks that are present as well. So the vaccination of mice is another potential valuable tool in our toolbox as we try to manage or reduce tick populations or the infection rates in these ticks. The oral vaccine is targeting the white-footed mice and probably chipmunks as well, one of the main reservoir hosts, although not the only ones. Some of the issues is that, what is the contributions of non-mouse reservoir hosts such as shrews? It doesn't target other tick-borne pathogens, so it's going to reduce the risk of Lyme disease, but not the risk of other diseases such as babesiosis and anaplasmosis, although there is some evidence that even reducing the prevalence of Lyme does have an influence on the prevalence of some of these pathogens in the reservoir hosts. And it, of course, requires, like any of these techniques that we're using, whether you're talking spraying or deer or whatever, it'll require annual ongoing treatment due to the mouse reproduction and population turnover. But nevertheless, without a human vaccine, the mouse vaccination actually ended up ranked second among modeled intervention options behind the acaricide applied to deer in preventing Lyme disease cases in some Lyme sim model simulations that were published back in 1999. And the vaccine is currently in a licensing process by U.S. Biologic, and you'll be hearing more about this a little later in the program, like I said. So here in Connecticut, what we're doing is looking at the ability of individual and integrated tick management technologies to reduce what we call the entomological risk of Lyme disease. In other words, the risk of encountering an infected Ixodes scapularis tick. And we're using 3 approaches in the town of Reading. The application of that MET52, the fungus, Metarhizium anopheles product. We're using the fipronil-based rodent bait boxes, and we're doing deer reduction as well. And we have now entering the 3rd year of the project, and not all the data has been completely analyzed at this point, but nevertheless, we're starting this. We've got about a 78% reduction in areas where we had the combination of the MET52 with the fipronil-based rodent bait boxes, which is quite promising. And we're also going to be adding into this intervention project the reservoir-targeted vaccine as a residential application. The original studies were done in woodland plots, so we want to see just how well it works around an actual residential setting as well. So what do we know so far? Where do we stand with all these interventions or strategies that we used? There's been studies that have been done showing that protective clothing and bathing within 2 hours after being outdoors is protective reduce ticks? Can it reduce Lyme disease? Yes. It's sort of indirect, but about 40%. Repellents, there's some evidence in surveys that have been done that it's about at least 20% effective. But interesting, a study that just came out, was just published, showed that only about half of residents at risk of tick-borne diseases are actually utilizing any form of protective measures. So there's still a lot of work that needs to be done in educating people, at least in terms of using protective methods, personal prevention measures to reduce their risk of ticks. Landscape modifications, we know that it has some impact on tick abundance. Does it reduce disease? That's unknown. Fencing, use is limited. Deer fencing is expensive. Can it reduce ticks? Yes. In the survey that was done, the case control study that found that bathing and protective clothing was protective, there was some evidence that fencing of some kind did have an impact on the incidence of Lyme disease in those in the study, but it's unclear how that was working in this case, because not all fencing was deer fencing. Chemical control with pyrethroids and the botanicals, yeah, we have data that they can reduce the number of ticks where they're applied. Can they actually have an impact on the incidence of disease? That's unknown at this time. Same thing applies with biological control using the fungus. It's a new market, so so we only have the studies to look at. It hasn't been widely used, obviously, at this time. But can it reduce ticks? Yes. Will it have an impact on Lyme disease or other tick-borne diseases? That's unknown. Host exclusion, deer fencing, that impacts tick abundance, but it hasn't been widely used. Does it influence the incidence of the disease? Don't know. Host reduction with deer, that has been only used in a number of communities that have achieved the necessary reductions to actually impact substantially tick abundance. And as I discussed in this presentation, we do have some data that it has impacted the incidence of Lyme disease, at least in a couple of cases. And with the host-targeted acaricides to mice, again, we don't know what the full impact of that is. It's been highly successful in some cases where it's used, but does it impact the incidence of disease? We don't know. Deer, host-targeted acaricides with deer, the 4 posters have seen somewhat limited use. It will reduce tick abundance, and at least in the one study, we know that it did have an impact on the incidence of Lyme disease. And lastly, of course, the host-targeted vaccine, the way that works, obviously it's not going to reduce tick abundance. It reduces the infection in those ticks. And this is brand new, of course, and so at this point we don't know what kind of impact it will have on Lyme disease or other tick-borne diseases, but conceivably it has a lot of potential to have an impact as well. So all of these strategies do have some use and have been shown to reduce ticks. We'll have to move forward to see whether it actually will reduce disease. So a lot of the strategies are summarized in my Tick Management Handbook, which is available on the Connecticut Agricultural Experiment Station's website. I'm going to be in the process over this next year of updating and revising the handbook, so you have that to look forward to. A lot of the basics don't change, but with new developments and new research, it definitely needs some updating in some regards. I've listed my contact information here, and that essentially concludes my talk for this morning.
Thank you, Dr. Stafford. We appreciate your work and appreciate you sharing that with all the residents in Dutchess County.
Well, you're more than welcome. So at this point, are we ready to move on to the next presenter?
I believe I'm going to talk for a few minutes while we get the next presenter ready to go, and then that presenter, which will be Dr. Eva Sapi, will then be with us. But thank you again, Dr. Stafford, and have a great day.
Okay, thank you very much.
For those of you attending our event today, this is Donna Bullner. I'm County Legislator for District 13 for Dutchess County, and I'm also the Tick Task Chairperson for our county. I'll spend just a few minutes here as we're waiting for Dr. Sapi to upload and come into our conversation here. I just want to talk about how we got to our webinar that we're having today. Last year, we actually presented in Salt Point, which is actually up in Pleasant Valley. We had 4 presenters last year, and we packed a firehouse with approximately 250 people. From that exact event, we were able to upload some videos that I myself videotaped, and obviously I'm not a really good videographer, but we were able to share those throughout the course of this year. And as I was thinking of how do we top last year's event, which was highly attended, I started looking at trying to really connect technology to communicating here with our residents. And so that's how the thought of coming up with a webinar really was developed, thanks to the people here at our Dutchess County OCIS working diligently on this, and our members of our Tick Task Force to help bring about some of our presenters for today. We were able to put together today 12 presenters rather than the 4 we had last year. We did allow 500 access codes to anyone in the county that wished to have one, and I think actually we have some access codes that are being utilized from out of state, which is wonderful. And what it will allow us to do is after today's event, we will be able to archive what we have for today, and we'll be able to share that continuing going forward, just like we shared the videos that we have from last year. So we're looking again at uploading this to our tab on the Dutchess County website. Anyone who enters the website would be asked to answer approximately, I think there are 6 to 7 questions that are helping us to collect data as we're moving forward, as we did from 2 years ago. It helps us to understand, you know, how tick-borne diseases are affecting our residents in our county, and also to help us move forward and know what resources that we need to have out there for our county residents. So again, like I said, this is really an effort of Carolyn Morris and Lee Wager out of our Dutchess County legislative offices. We have members here of OCIS that have been working diligently on this. And again, our Tick Task Force members really helped us put together, I think, a great webinar with terrific presenters. And as you just saw from Dr. Stafford, there's a lot of information that is very technical in nature, but at the same time, it's information that I think our residents here in Dutchess County can actually use immediately and start to think about. And remember that at the end of each presentation, our presenters are putting up a slide that will allow you to direct any questions to them. However, if they do not have a slide, you can direct your questions to DC, that's D as in dog, C as in Charlie, Tick-Borne at dutchessny.gov. And again, if you send your questions directly to that website, we will get them answered and get an answer directly back to you. And of course, you know, like I said, a shout out really goes to everyone in our county. It is May. We did proclaim this month to be Lyme Disease Awareness Month. We want everyone to think of those protective acts, especially that Dr. Stafford talked about, especially in the clothing area. And watching, you know, making sure you're doing tick checks when you're coming in, and especially for our children. And we'll talk a little bit about including some students in the conversation of tick-borne diseases here in Dutchess County a little bit later. So at this time, I'd like to introduce Dr. Eva Sapi from the University of New Haven. And Dr. Sapi, if you want to introduce your front slide, that would be wonderful.
Thank you, Dana, and thank you, Dr. Stafford, for the beautiful presentation about, you know, tick control. And again, my name is Eva Sapi, University of New Haven, and I was back and forth what to present today, and I really felt very strong about that we need to understand better antibiotic resistance of Borrelia biofilm. That is exactly why we need those very serious tick control, because there are some issues. So let me talk about those. What are those issues? And I always keep this slide because I always want to remind everybody that it was in 1982 when Dr. Borrelia discovered spirochetes in isotonics, and that is over 30 years ago, and we're still struggling with Lyme disease. And very soon after, he showed that the spirochete reacted with immune serum from the patient with Lyme disease. So again, 30 years, and we still don't really know how to eliminate this organism. And again, obviously last year it was a big, the announcement that we have over 300,000 people who are affected by Lyme disease, which is a very significant number. My students always ask me, so how long do we know about Lyme disease? And again, just recently we realized that this bacteria is around more than 50 million years. We even found some DNA evidence in Ötzi the Iceman, who is over 5,000 years old and is the first Lyme disease patient. So obviously the question is, how can we eliminate Borrelia? When I looked at studies on lab studies about the drugs, antibiotics, which are effective against Borrelia, actually there are at least a very good effective agents available according to those lab studies. But are they effective? And if you look at the list of these papers, it's back in 1989 when we already seen survival of Borrelia in patients who were treated with antibiotics. And since then, we have more and more clinical data that something is not right. Of course, you could say, why not just using mouse studies and testing those antibiotics in a mouse system? And even in a mouse study, back in 1994, we already seen that somehow something is surviving, something is not responding to our treatment. Yes, but you're going to say, yes, but we have ceftriaxone, which is phosphine, and this should work against Borrelia. And again, I have to remind you, as early as 2002, they already showed, studies already showed that there's something surviving the treatment. Especially Dr. Bokenstein and Dr. Hartig, Dr. Barthold studies show that it is a non-culturable spirochete which survives, but this non-culturable spirochete survives a treatment. What they did, they even took out a piece of tissue from the treated mice, put it in a second mice, and they put a tick on the second mice, and the tick got infected. So something surviving for treatment. Of course, a lot of speculation at this point. What's happening here? What is the mechanism that this spirochete can survive treatment? And this is one of the interesting picture showing Borrelia sort of coiling up after penicillin treatment. So a lot of people started to question it, what's happening to Borrelia after we add antibiotics? And I would like to remind you that we have, when you have Borrelia in the culture tube, this is the culture tube in our laboratory, you see spirochete, beautiful spirochete, but you always see those little dots. And of course, you're going to see probably some dirt in the culture. And the reason I know it's not a dirt because this Borrelia is actually labeled with a green fluorescent protein. So those dots actually fluorescent, so we know it's Borrelia. We call it cysts or granules or round bodies. Dr. McDonald also talks about a form which is called a cell deficient form, and he sees this even in tissues. So, and today I'm gonna focusing on something we discovered not long ago called the biofilm form. So how about the questions? How can we eliminate those different forms? First of all, are they real? One of the studies back in 1988 when Dr. McDonald looked at those different forms in cultured hippocampus from Alzheimer's patient, and he sees those round bodies on this tissue. But there are other studies which also proven that those forms are indeed real, and they can find it in the different tissues. This is Dr. Grunter 2001 study. So can we have some agent for those forms? Obviously, that was a big question, and Dr. Bronson from Europe spent a decade to finding different agents like metronidazole, which is flagyl, or he has some other agents which he found it might be effective against those round bodies. One of the agents, the tindazole, actually we used in our study also, and we also confirmed it's very effective against those round bodies. The last paper he published was back in 2009 when he used tigacycline, and he said that tigacycline is extremely effective against those spirochetal round bodies. So the question is it working in vivo? And unfortunately, back in 2010, Dr. Barker repeated the tigacycline experiment in his mouse model, and unfortunately, same as before, he found that it's something against surviving those treatments. Dr. Embers back in 2012 also seen but Borrelia can persist after 90 days in monkeys who treated with different antibiotics. This is her picture. Here is practically the tick, which was tick midgut, which placed on the treated animal. As you see, is plenty spirochete in the tick midgut, even some interesting forms. And this is Borrelia in a treated animal. So definitely something survived the treatment. So, and the latest one, which I think the most convincing evidence, is Dr. Hurtig's data when he showed that the treated animals are showing some signs that Borrelia DNA can survive the treatment. So what happened is he used PCR methodology, and he monitored the treated mice after 2, 4, and 8 months, and he saw that a decline in the DNA, which makes sense because you expect that that the treatment works. However, there was a significant increase in spirochete DNA load in multiple tissues at 12 months. It shows that something surviving, something is still feeding this DNA load. And here's his paper. As you see, again, this is Borrelia after 12 months after the treatment in the ticks midgut. So something surviving, and of course the question, what can we do now? And I always show this 2008 data when we looked at different antibiotic treatment on spirochete. Here is the control, and here is penicillin, doxycycline, and we sort of looked at under the microscope what's happening to the culture. And as you see a lot of cysts, which we expected, but we also seen something covering those cysts. And we didn't understand, and we thought maybe some contamination. And we talked to Dr. McDonald at this point, and he showed us this paper, Dr. Eisendahl from Austria, who sees something similar in infected skin tissues. So we got very interested, and we started to look at some other tissues. And as you see this back in Dr. Ember's monkey studies, remember I showed you that it's again something that tick midgut, which is not just spirochete, but looks like something covering. And this was the picture Dr. McDonald sent us to back in 2008, and he showed that it's something between those two Borrelia aggregates, and looks like it's almost like a bridge and looks like a communication. And that's when we suggested that Borrelia can make something called biofilms. So what is biofilm? Biofilm is a collection of microorganisms. And the good example, plaque that forms on the teeth if you don't clean it, or the slime that forms on a surface in a showery, watery areas like the shower. The slimy substance is extremely important because it's a protective layer for the organism. And the most important thing about biofilm that demonstrates very high antibiotic resistance, it could be high as 1,000-fold. This is a major issue in clinic, especially on those medical devices. It can get infected, and unfortunately, those infections doesn't look like it's responding to regular antimicrobial treatments. Here's just a couple organisms. This is a couple examples for biofilms, from dental plaque to middle ear infection to urinary tract infection, even infectious kidney stones. This is a picture about dental plaque, and if that doesn't take you to the dentist, nothing will. So we have to prove at this point that Borrelia can make indeed biofilm. So we looked at the literature, how can we prove this? And we found a couple of criteria, and we actually focused on two major ones. One is those internal morphological rearrangement, because in biofilm it looks like a city, and when in a city, what is the most important thing? Bring the food in and take the waste out, and for that one you need channels. So we were looking for those channels in those aggregates. We also were looking for the slime, because that's also a very characteristic for biofilm. And we also have some data for communication. We don't have time today, but maybe another time I talk about it. So this is Borrelia gathering. As you see, you can see very clearly still the spirochetes. But a later stage, as you see, the inside you cannot really see the spirochete, but if you see the side of the biofilm, you can still see the spirochete, how it is affecting and building the biofilm. And here is the three stages of biofilm development: an early stage, a mid-phase stage, and the later stage. This is two different microscopy methods. I want to show you this one because this structure, I remembered when we looked at this structure, actually is even crunched, which was very scary at this point. And I'll give you some explanation why it could be so crunchy. And as you see, without a very high magnification, we couldn't really look inside the biofilm, so we needed to use a new methodology called the atomic force microscopy, which is able to see live biofilm development under the microscope with nanoscale magnification. And here's the very first image we got. As you see, here's the spirochete. As you see, they start together, and as you see, something looks like a flagella, which should be inside, but in this case looks like outside. It might be happening the development. And I'm going to show you quickly couple images, so you will see how the biofilm forming in a very high magnification. This is exactly the same slide. We monitored it for 2 weeks. And we were seeing that it's definitely a very organized structure building here. This is my favorite picture. Looks like a crochet. And as you see, as we waited a little bit longer, the symmetry is a little bit disappearing, but what we're seeing is the channels forming. And this was the first time we were started to believe that we're really looking at the biofilm formation. Here is a composite image for several scans. As you see very clearly, you can see those channels. This is just look inside the channels. And here something, even see some kind of protrusion. We still don't know what does it mean. We're still working on it. This is another picture. And this is just a composite picture. So we definitely proven that it's a very significant formation of biofilm. So the next thing is the slime. How can we prove it is slime there? The slime actually is a main component of the slime is mucopolysaccharides, which is basically sugars, but also has some other components. So here's it's also an unspecific staining for slime. As you see, very strongly stained. The two different colors represent two different classes of mucopolysaccharides. So we really wanted to know what is this slime exactly, and one of the things everybody is looking at this point is alginate. And so we got some antibody from Dr. Pierce, Harvard, and we actually were able to show that alginate indeed forming on Borrelia biofilm, but not forming on spirochete. This is a picture from a paper. Alginate binds to calcium, And as you see, remember I mentioned that it was this little structure was very crunchy. We found tremendous amount of calcium staining on those biofilms. I also mentioned there are other things protecting the biofilms. One is we call the extracellular DNA, and again we've proven that extracellular DNA is on the biofilms. We're still looking for additional protective material. I have a student who's looking for defense lectins, acetylglucosamines. This is under investigation right now. So of course you're going to ask me, so yes, that's in your lab, in your test tube, but how about in real tissues, infected tissues? And I don't have time today to talk about this study. I will show you one image. This is in infected skin tissues, and we already have some very strong evidence that indeed this biofilm formation is not just in a test tube, but in the tissues. Of course, the question, however, still remains: how can we eliminate Borrelia? And I always show this picture, and you can see Borrelia biofilm in the test tube treated with high dose of doxycycline for 3 weeks. What you see on the left side, it is a red aggregate. Red means it's dead because we used a specific staining. But however, the green means it's still alive. And as you see, it's plenty, another colony aggregate right there, which is staining very strongly with green. And this was the very first study we done trying to see what kind of antibiotic combination is effective against biofilm. And even the best combination, as you see, maybe 60-65% effective, but it's still not 100% effective. Here's just a picture. Sorry, here's just a picture of the same study. Looks like doxycycline and thiendazole is effective, and we were very happy about it until we repeated the experiment with different antibiotics similar to thiendazole, but this time we let the biofilm actually sit on collagen. When we let the biofilm sit on collagen, unfortunately, the result didn't look as effective. And we unfortunately see this all the time. This is another study when we compared antibiotic sensitivity, when we looking at plastic as a surface for biofilm or collagen. EDTA is a chelating effect. Remember, it was a lot of calcium, so we were wondering maybe EDTA would be effective. It was effective when it was sitting on plastic, but not on collagen. And unfortunately, our studies, or in vivo studies, already shows that Borrelia in the body is very, very. We can find them in the collagen-embedded tissues. We try different enzymes. Everybody always asks me, did you try this enzyme, this enzyme? Unfortunately, not that many things is working against, especially when it's sitting in collagen. We tried even another one, DNase one. It's just not effective. So just recently, John Hopkins showed a very interesting study when they screened over 15,000 different antimicrobials from an FDA-approved library, and they find that none of the agent as a single agent able to eliminate. They call the persisters. Persisters in our laboratory, the persisters are those biofilms which is basically floating, not on a surface. As a single agent, actually was daptomycin, which was most effective. And in the latest study, actually they said it is only a combination, doxycycline, daptomycin, and ceftriaxone, which was effective eliminate those persisters, Borrelia persisters. So obviously at this point, we decided we might need a Trojan horse here, because it's somehow we need some better approach to eliminate Borrelia. And the whole idea came from a study from 2011 when they were going after persisters with sugar and some antibiotics. We found the study very interesting, and I had a student who practically tried every single sugar available on a market. And interestingly, she found something that maybe something called stevia, which you know as a steve leaves, and you might even use it in your coffee or tea. She found it very effective against Borrelia, especially Borrelia biofilms. And this is just a very recent study which we're publishing right now, showing that doxycycline, ceftriaxone, and daptomycin, as it's reported by John Hopkins scientists, are very effective against log phase and stationary phase as stevia. I don't have time to talk about jalapa, maybe next time, as stevia. But when we repeated the same study on those attached biofilm I mentioned to you, looks like stevia was as a single agent was outperformed the triple antibiotic agent for Borrelia. And here's just a study, which again we hopefully send in the next couple of weeks. As you see, stevia as a single agent, again the red represents that biofilm performed the best among all those agents. As I mentioned, we have atomic force microscopy, and if you see, we scanned those biofilms after different antibiotic treatment, and again, stevia produced this very interesting, very loose structure, which hopefully represent. Again, we need more studies. Hopefully, represent an effective treatment. So in summary, Borrelia burgdorferi does form a very organized biofilm in vitro and in vivo. The major surface, the protective surface, is calcium alginate and different lectins as well as extracellular DNA. Borrelia biofilm does provide a refuge from different antimicrobial treatments, and looks like we are able to identify some few potential agents. Today I didn't have time to talk all about the other agents we found, but so far stevia, one of the leading agents we found for Borrelia biofilm. This is just a picture of my lab. You can see the atomic force microscopy. You see a lot of results from this experiment. This is just a sort of a list of my students who finished the last now 11 years as a thesis project or a research project. And I have a couple of thank you slides. Always University of New Haven, College of Arts and Sciences, all those organizations who helped us funding this study which you've seen. And of course, all the state-of-the-art microscope, which is essential for our studies. And of course, my department, my chair, who always protecting us and always providing a nice, safe environment for our studies, and Dr. McDonald for his never-ending support. And here's my slide, contact slide. Please call me or send me email if you have any questions. And thank you very much for inviting me. I hope you enjoyed the presentation. Thank you.
Thank you to Dr. Sapi. At this time, we will be moving to Dr. Thomas Mather. And Dr. Mather, if you don't mind just introducing yourself and your title and beginning your presentation.
Okay. Hi, I'm Dr. Tom Mather. Sometimes people just call me the tick guy. I've been focused on ticks and solving the tick problem since 1983, different aspects of it. And one of the things that we have done that I'm going to talk about today is build and try to maintain as best as we can a tick bite prevention resource to help people with making appropriate decisions, basically translating the great science of people like Kirby and others that you'll hear today, so that it really meshes with people's lived experiences. And I think you'll be interested to see some of the work that we have. I'm giving you an update today on one of our tools called Tick Spotters. It's a crowdsourced tick survey for America. So I've got to figure out how to change my slide. All right, sorry for the slow start here. I believe that we have more ticks in more places now than ever before in probably the history of North America, and part of that is related to the changing landscapes that we have here. The landscapes have changed so that there actually are more adapted to the hosts that serve as reproductive hosts for black-legged ticks in particular, but many other species as well. But I don't imagine there's anyone from Dutchess County or other places that would disagree with the fact that there's more ticks now than they've ever remembered in the past. And so that's a problem. In fact, it's more than a problem. It's become a public health crisis for communities throughout America, not just deer ticks and Lyme disease, but other ticks and the diseases that they carry as well. And so in this more ticks in more places landscape, there's more information, lots of great science going on, but there's also an extraordinary amount of misinformation out there for people to try and wade through when it comes to dealing with their tick problems. And so that was a gap that we felt needed to be addressed, and that is, how do we try to focus the information into resources that people can trust, that it's based on science rather than hearsay or anecdote. In fact, I believe that America suffers from a tick literacy deficit, and that this tick literacy deficit is holding us back from really focusing on the best next actions to take to protect ourselves from tick-borne diseases. So one goal of our program with TickEncounter is to try and work to erase tick literacy deficit, and I'm going to tell you a little bit more about that as we go along. So where did the tick literacy deficit problem come from? And I would argue it comes from back in the day, as my son likes to say. Back in the day, we all had ticks. Everyone had ticks. Ticks like this, American dog ticks, climbed to the top of your head, rarely made anybody sick. And you know, your mom was probably the one that pulled it out because she had pointy fingernails like tweezers. No one had tweezers back in the day, really, and pulled this tick out. But since it rarely made anybody sick, generally people just got rid of the ticks. There was no real public health crisis. Yes, there were some cases of Rocky Mountain spotted fever, but not so many. And the ticks were just dispatched and out of sight, sort of out of mind, till the next one came along. These days, however, I think the situation is quite different. There's a new tick, not really so new now, 30, 40 years, but it's increasingly affecting people coming into their own backyards. And this tick comes in small, medium, and large sizes, just like all ticks, but the tiny stage of this tick, the nymphal stage, is a very potent vector of multiple pathogens. So it's basically like getting a new game. When you get a new game, you kind of need to read the rules if you're going to know how to play the game. And so we are basically at that point where new rules need to be written and need to be understood by people, because this tick is loaded with pathogens. It doesn't really crawl to the top of your head. And so already there we see that there are different things going on with this tick compared to the American dog tick. So this is just to give you an idea of some of the tools on our TickEncounter resource webpage. We have the new rules right here, the top 10 things everyone needs to know about ticks these days. You can look at that yourself. It's under this prevention tab if you wanted to go to TickEncounter and click on that and look for the top 10 things that you need to know these days if you're going to try and stay tick safe. So our issue is that we're providing information, but in order for people to actually be willing to take action, it's all about a health promotion program. So in order for health promotion to be effective, people basically need to believe that they're susceptible and likely to fall victim to a disease. And I'm imagining that a lot of people in Dutchess County and elsewhere living in tick endemic areas do believe that they're susceptible, but I don't know that it's always someone else that's going to get it. They may think they're a little bit immune. So that's a little bit of an issue right there. But they also have to believe that the disease is going to cause them significant adverse consequences. And while all of these horror stories are out there, there is still not a strong belief that people actually are going to get all that sick themselves. They have to believe that adopting any preventive behavior is actually effective. And, some of the studies that Kirby was mentioning earlier, and there are many more studies out there where we're looking at evidence-based studies that actually show that taking any preventive behavior is effective is going to be critical for helping people along this. Finally, people have to believe that they can actually do it, and this is where the whole translation piece comes in. You can read about it, you can hear people telling you strategies and everything, but until you feel that you actually can do something, then it's not going to happen. So you have to be empowered, which is what we think is really an important key to an education process here. So our goal with some of our programming is to develop community-driven public health strategy for solving America's tick problem, changing people from misinformation to talking about tick-smart actions that they actually can do and that they do on a regular basis. That would be our goal. We have 5 core tick-smart actions on our as part of our program. You can see these here. We're going to focus today on this one called Know. We have to know the kinds of ticks active throughout the year, when they're active, so that we know how best to protect ourselves. And certainly when we encounter a tick, we need to identify it correctly so that we do the next best thing in managing that tick bite, and we can assess our risk. So we're going to focus on that for the rest of today's presentation. I like to start right here. Social media can be good and fun, but it can also be a little bit contributor to the tick literacy deficit. But this is a great example. This woman had this picture of a tick. Everybody probably watching knows exactly what kind of tick that is. And she reached out. She found it behind her granddaughter's ear, so I'm sure she was panicked. And so here's a little bit closer picture. And you can see that her friends tried to help her, and you can just look at some of their comments, slightly misguided, as you can see by these emoticons over here, a little aghast myself as to what people know and believe and are willing to share. So this one looks too big to this woman to be a deer tick. She forgot the top 10 things that you need to know, which is all ticks come in small, medium, and large size, for instance. And so once we finally got into this conversation, we were able to provide her with, the correct ID and maybe even a little bit of a risk assessment, because this tick was clearly attached for at least 2.5 days. And since about half of these adult deer ticks are infected, she might really want to have this tick tested in order to better assess the risk from this tick on her tiny granddaughter. So that would be the type of strategy that we'd like people to take, and we've tried to build resources on TickEncounter. This is one of our more popular pages. It's a tick identification chart where on your screen it tries to give you sort of actual size pictures, and then if you click on any one of them, you can see more of a magnified size. If you click on that, you actually get supersized looking pictures, and you can flip them over top and bottom, and you get more information as well. So this is under the tick identification tab on TickEncounter, and you can see here that it's a different chart for different regions of the country, so highly engageable. We realize that those ticks often don't look like the unfed tick that everyone has on their tick identification pages, that they actually change their appearance as they feed, and you're more likely to see this and not necessarily be able to recognize it as this. And so we've built tools that help people see ticks in different stages as well. We've also developed tools like tick identification guides. You know, when you're looking at a great big engorged tick, most people see the great big engorged tick, but if you have any hope of identifying what it is, you need to focus on what's unique to that tick. And so the scutum and the mouth parts are unique to each type of tick, at least in the adult stage. And so if focusing on that, you can start to see the differences where most people's eyes just see a great big yucky tick. So why is it so important to be able to identify ticks? Part of it is because different ticks, the different species of ticks that we're likely to encounter here in your area or along the eastern United States in particular, carry different diseases. And they don't really overlap all that much, so I don't have time to go into this, but it's important that the motivating factor is knowing what type of tick can help you assess the risk for various diseases. So we've been building tools to try and extend the reach. This is the beginning. This would be part of something that could be locally branded, let's say by Dutchess County even. They could put their logo here. It would look like they have, really fine and sound tick literacy information. Our process that we'd like people to follow when they find a tick is that they would save it, not just pitch it. They'd try and take a nice clear picture. They'd maybe use some of our identification tools to identify it, and then maybe even submit it to our crowdsourced Tick Spotters program, where we can validate the tick ID and also give best next actions to take. So this is a tool that we're rolling out to partners right now. We call it the Tick Smart Web Portal, something that would be available to communities. And here's the Tick Spotters program part of that. So you can see there's a tab for Tick Spotters right there. And the whole point of this is to assess the risk and guide next best actions after finding a tick. So this is what a submission report looks like. So you can see here's one from New York. This is from up near Niagara Falls. Get the date. You can see the picture of the tick. Sometimes they're not great, but from this picture you can clearly see it's an American dog tick, but the person didn't know. And so they might have assumed that it was a black-legged tick and they were going to get Lyme disease, and our reply then can show them that that's not their issue. So this is what our current reply in an email looks like now, very customized for their particular case with hyperlinks to Tick Smart actions to take. This is what we'd love the report to look like eventually once we get some funding. Nice professional looking thing, still with the same hyperlinks. And then one of the things that can be recommended would be tick testing. And so we have a new partnership, a two-step process with UMass, where Tick Spotters to get the risk assessment and tick report at UMass to have your tick tested to see if it was actually carrying any infections. These are just some of the 2014 results that we've gotten from our Tick Spotters program. You can see these are heat maps to show where tick activity, this is all ticks coming in from Tick Spotters in the winter. I'm just showing you these maps to show you how the patterns change a little bit. In the springtime, there's a little bit different pattern. In the summer, again, it changes a little bit. So just shows you where the ticky areas of the United States are. You can see in the fall, it's pretty high high-powered up here in the northeastern United States because of the black-legged tick adults. We'd like to turn this into an informative dashboard that gives people information. So focusing on last spring, since it's this spring, we don't have this data in real time. Again, here was the heat map. This is the distribution. You can see that 16% of the people submitting a report didn't actually know what type of tick it was. Actually, I think that number is even higher, and we actually looked at pictures and corrected that. So maybe this is what we couldn't even come up with because maybe the picture wasn't so good. You can see here sort of a trend that springtime is the tickiest time of the year across America. This is based on about 10,000 responses to this program last year. You can see that we have pictures, real people's pictures. This one came from Michigan. Again, trying to keep ticks on the top of people's minds, sort of what's going on in that season. So seasonal relevance is really important. Here's another one from Michigan. Here's another one. This is a male Lone Star tick. Also, you might think Michigan had the most ticks, but that's just the way it turned out with the pictures that I selected, I guess. Just narrowing it down, you can see these are based on lots of points. These are points from the spring of last year in the Northeast region. And you can even focus in, this is your Dutchess County area right here. And these are zip codes. This is in this particular zip code, 12525. Maybe some of you live there. And this just shows that the individual records of the submissions, these are placed in the center of each zip code, but this one, for instance, there were 6 submissions from that zip code during 2014. So getting more information eventually available to people, I think, is going to be helpful in motivating them to take next best actions. This is just some summaries currently. So in April, you could see how as the snow melted, we started to get many more reports. American dog ticks and black-legged ticks were kind of the most commonly reported, except for the I don't knows were far more common here. Again, this tick spotter reports gives us an unending wealth of people's lived experiences with ticks. We often can then turn them into social media posts. Here's a great one, you know. People always say don't burn a tick off. It's never a good idea to do it, but I actually had never seen someone submit a picture like that, so that's kind of interesting. Here is here's for May. We're in our current situation. It's ramping up considerably, you can see, and the American dog ticks are actually becoming far more abundant. This particular post was a pretty good one. We put a public health message to it, put it out last Saturday, and it ended up so far, as of last night when I put this together, 76,000 Facebook views. So you can see that Tick Spotters has a lot of opportunities for helping drive the goal and the effort to erase tick literacy deficit. You're welcome to contact me through TickEncounter. We have several different ways. There's a www.tickencounter.org/contact email service that would get information to me if you had questions. Thanks very much.
Thank you, Dr. Mather. We appreciate you taking the time out today to share this information with our Dutchess County residents. Again, this is Donna Bollner. I'm the Dutchess County legislator, but I'm also the chairperson of our Tick Task Force here in Dutchess County. I spoke to you a little bit earlier this morning in regards to the development of this educational webinar, where we were last year and how we got to this point this year. Right now I'd like to talk a little bit about the youth that we have and some things that we've had going on here in Dutchess County. Last Monday night on May 11th, we actually gave awards to our 2 poster contest winners. Our Be Tick-Free poster contest, which was developed out of the Dutchess County Health Department. Its history is that in past years, our federal government had sponsored a similar poster contest for 5th and 6th graders, and we participated as a county and actually had several of our students in our county go to the state level on that particular contest. And when the federal government discontinued that, we decided that it was important at the Tick Task Force to continue that because we thought the education piece and connecting with our younger generation, because they are the next generation that will, or actually they're the current generation. If you look back to the information we got from Kirby Stafford this morning, those are the students that are out playing. So developing some sort of a proactive approach and allowing them to have a contest was something that we felt that even though it wasn't going to be sponsored anymore through the federal government, that we would sponsor it here in Dutchess County. And like I said, it is in conjunction with the health department. There we have a nice relationship with Dr. Cary, who's the commissioner there, Andrew Rotans, and Aisha Phillips. Kudos go out to her because she really has taken over this poster contest that was started actually by Kristen Herzog, who's now in a different county department. So our 2015 poster winners this year were Nathan Ogden and also Brandon Chin. Nathan's from LaGrange, where I actually reside and represent a part of, and Brendan Chin is from, I'm sorry, I'll take that back. It's actually Brandon Chin, not Brendan, but Brandon Chin from Fishkill. Were our 2 winners this year that developed posters. And like I said, we honored them Monday night in legislative chambers during our board meeting for their efforts, and we look forward to more poster contests in future years. Also, we hope to develop a relationship. Nathan actually reached out and would be willing to help us on the Tick Task Force. As an educator for 20 years myself, I think it's important to reach out to our youth and to kind of understand Lyme disease or tick-borne disease through their eyes, because sometimes seeing it through a child's eyes will actually help you to develop better ways to reach out to those. And again, from Dr. Kirby Stafford's presentation this morning, they are a age range that is at great risk of tick-borne disease here in our county. Also, on the flip side, our county does have we did lose a youth. Joseph Alone, who was a Poughkeepsie High School student, did lose his life to a tick-borne related illness. And Joseph was one of those students that was in environmental studies, hope to go forward and do more in the environment. Obviously, he spent a lot of time outside and contracted and developed a tick-borne disease. But again, I think Joseph's life and in his love of nature and his love of the things why we reside in Dutchess County are to be commended. I know on June 8th at 7 p.m. at the Vector-Borne, there is going to be a presentation called The Little Things, and it will be with his family talking about his life. And so like I said, we are again, we reach out and think of Joseph as a student here and a person why we should continue to fight tick-borne diseases here in Dutchess County. And we lift up Nathan and Brandon as that future. As we come to our next presenter, which will be Richard Ostfeld from our own Cary Institute here in Dutchess County. I again just want to thank Grant Haight, Richard Byrne, and Deborah Campion for all of their work to make this possible today. This is, I think, a true gift to Dutchess County, and obviously it will be archived, and many other people, even from outside of Dutchess County, will be able to view it. So with that, I'd like to welcome Richard Ostfeld.
Thank you, Donna. I will assume, unless someone tells me otherwise, that I can be heard loud and clear. I've been kindly given 2 20-minute slots. So what I would like to do is give a research update that I'll break down into two parts with a very brief, about 5-second long break in between at approximately 20 minutes from now. So as I said, this is a research presentation, and it is directed particularly with regard to tick-borne diseases in the Hudson Valley, and I will be discussing the ecology of risk. And there we go, to advance the slide. Okay, so Dutchess County, as is true of various other parts of the United States and other parts of the world, has experienced wave after wave of the emergence and spread of diseases that are transmitted by black-legged ticks. So back in the 1980s, we were exposed initially to Lyme disease, which was first described only a few years earlier and has been spreading ever since. Then about 10 years later, maybe a little longer, we saw the emergence and spread of another bacterial disease. I should say, I'm sorry, that Lyme disease is caused by this spirochete bacterium, Borrelia burgdorferi. The next wave was caused also by a bacterium, although not closely related to Borrelia burgdorferi. This one is a rickettsia-like bacterium called Anaplasma phagocytophilum, transmitted by the same ticks that emerged in the 90s. And then somewhat later than that, we saw the emergence of a new disease called Babesia microti, transmitted by a protozoan parasite related to the malaria parasite in the 2000s. Now, more recently, we may be seeing the emergence of yet another disease. It's not yet clear, but this is caused by another spirochete bacterium, Borrelia miyamotoi, a relative of the Lyme disease spirochete, but in a different group that tends to cause a relapsing fever. This is fairly newly described in Dutchess County and a few other places in the northeastern U.S. It has no particular name for this disease, so I'm calling it B. miyamotoi infection. Whether or not it's going to emerge, we don't really know. It is present, and I have little, if anything else, to say about it in this presentation this morning. What I'm showing here are data on the actual reported numbers of cases for each of these three diseases that I'm going to focus on right now. Lyme disease, as I said, was first described in the 1970s and first became a notifiable disease in the early 1980s. Early during this emergence, there were only a few hundred cases reported in the United States per year. And as you can see from this slide, the number of reported cases of Lyme disease has been growing inexorably throughout the time period over which the CDC has been recording cases, such that in the last few years there have been in the tens of thousands of reported cases of Lyme disease throughout the United States. Last year, the CDC made an attempt, a rigorous attempt, to estimate the degree to which these reported cases are actually underrepresenting the actual number of cases of Lyme in the U.S. And they have estimated that the reported cases should be multiplied by 10 to get a decent estimate of the actual number of cases. So in actuality, there are on the order of a few 100,000 cases of Lyme disease, new cases, reported in the United States every year. For human babesiosis, this was only made a notifiable disease a couple of years ago, and so there are not any good national statistics showing trends, but these data were kindly provided to me by the New York State Department of Health. This is for New York State alone, and again, we see this inexorable increase in the number of reported cases of babesiosis in New York. For anaplasmosis, we see the similar pattern, although even perhaps more alarming. The number of cases looks like it might even be increasing exponentially over recent years. And I just remind you that all three of these diseases are transmitted solely by the bite of a black-legged tick. So tick-borne disease in the 21st century, this is true in Dutchess County as well as elsewhere in the world, is a very vexing problem. As I've just shown you, there are several of these diseases that are rapidly increasing in incidence. In no case is there a vaccine available to protect humans against infection. Diagnosis and treatment for all these diseases are problematic. They are highly controversial. Recent estimates of the healthcare costs in the United States for Lyme disease alone range from about $712 million to about $1.3 billion a year. One thing that is fairly uncontroversial that almost everyone agrees on is that prevention and tick management or risk management are absolutely key. If we can avoid problems of diagnosis and treatment, we can avoid some of these massive healthcare costs if we can do a better job of preventing tick bites and transmission of these diseases to people. Fundamentally, prevention and tick management or risk management require an ecological understanding. We need to know where and when the ticks are most abundant and most heavily infected, where we're most likely to encounter them. That requires an understanding of the ecology of the ticks, their hosts, and their pathogens. And that's what I devote a fair bit of time, as well as some of the other speakers this morning, to trying to determine. So what I'm showing here are the three post-egg life stages in the life cycle of the tick. Black-legged tick, Ixodes scapularis. Tom Mather also emphasized that there's the small, medium, and large. This is the larval tick. This is the nymph, and this is an adult female. All three of these stages take a single blood meal from some animal host, some vertebrate host. That blood meal lasts anywhere from 3 or 4 days to about a week or slightly more. The tick, after this several-day-long blood meal, drops off the host and molts into the next stage, larva into nymph, nymph into adult, and after the adult's blood meal, the female drops off, reproduces, and dies. So there are a couple of very important features of the tick life cycle that are relevant to understanding our risk of getting exposed to these tick-borne diseases. The first is that these larval ticks hatch free of infection with any of those three tick-borne pathogens that I've just described, the agents of Lyme disease, babesiosis, and anaplasmosis. So these ticks, so in the scientific parlance, there is no trans-ovarial transmission. Mother ticks do not transmit any infection they might have into their babies, and so these larval ticks hatch free of infection. But it turns out these larvae are what we call extreme host generalists, and what that means is that they will readily feed off of almost any warm-blooded animal that happens to come by close enough for them to grab a hold. Almost any mammal or bird is a good target for their feeding. If they happen to feed on an infected host, they might acquire an infection with any of those three tick-borne pathogens, in which case they will molt into an infected nymph. So the second key feature of the life cycle is that pathogens must be acquired from vertebrate hosts, and it turns out that this larval blood meal is a critical one because it will determine whether the larva acquires a pathogen and molts into an infected nymph. And we know from overwhelming epidemiologic data that nymphs are responsible for transmitting the vast majority of tick-borne infections to people. So when it comes to ecological risk, where it comes from, where it's worse, what to do about it, what we need to know are two things. How many nymphs are there out there crawling around in the woods where people might encounter them, and how many of those nymphs are infected with one or more of these tick-borne pathogens. So in my lab, we've spent considerable effort addressing this question: which animal hosts are responsible for infecting ticks with these three pathogens? In other words, which are what we call the reservoirs for infection? And we do this on and near the grounds of the Cary Institute in Millbrook, right smack in the middle of Dutchess County, New York. We do this by going out in August, which is the time of year when larval ticks are actively seeking a host, and we go out and capture every single animal we can possibly get our hands on. And this includes things like woodchucks, shown here with a lovely salad bar. This is a southern flying squirrel. We catch long-tailed weasels, and this, of course, is a raccoon. In all cases, what we do is we hold these animals in cages, feeding them abundantly and giving them plenty of water and nesting material in some cases. And the cages have a wire mesh bottom that allows anything on or in the animal to drop off and be retrieved by us in a pan that's held under the cage of all these animals. This allows us to collect all the ticks that are naturally feeding on these animals, and then at the end of about a week-long period, we release these animals back at the place where we captured them. So what we have found out is which of these hosts are most responsible for transmitting infection to these feeding larval ticks. So what I'm showing you here are the data for Borrelia burgdorferi, the agent of Lyme disease. What we have found is that there's huge variation in the probability that each animal host species will transmit an infection to these feeding larval ticks. 92% of the ticks feeding on a wild white-footed mouse in and around Millbrook, New York, acquire an infection with the Lyme disease agent and molt into an infected nymph. And that's the highest value that we get. There are a few other hosts like chipmunks and 2 species of shrews that are moderately good reservoirs, but most of these other species are really quite poor at transmitting any infection that they might have to feeding ticks, and that includes squirrels, ground-dwelling birds, skunks, opossums, raccoons, and deer. And in fact, make note for a second that 3 of the 4 most competent reservoirs for the agent of Lyme disease are the white-footed mouse, the chipmunk, and the short-tailed shrew. These are the data for Babesia microti. Once again, we find that white-footed mice, short-tailed shrews, and eastern chipmunks are 3 of the top 4 hosts when it comes to reservoir competence. It also turns out that raccoons are a good reservoir. So here are the 3 small mammals that are highly efficient reservoirs of infection for Babesia. But I'm graying out the bars for raccoons and opossums because we found from these hosts a genetically distinct strain of Babesia microti that has not yet been found in human patients, so we're not quite sure what to make of this relatively high reservoir competence for raccoons. And in the case of Anaplasma phagocytophilum, once again, very large degree of variation among hosts in the probability that they'll transmit an infection to a feeding larval tick. But once again, we find that the white-footed mouse, the chipmunk, and the short-tailed shrew are the 3 most competent reservoirs, infecting more of the ticks than is the case for any of these other hosts. So what I've described here is a pattern. There's a clear pattern that the same group of small mammals are the best reservoirs for all 3 of these tick-borne pathogens. White-footed mice are always the best, and then we see chipmunks or short-tailed shrews as being the 2nd or 3rd or 4th best in each case. Well, so that's an interesting observation, but so what? What impact might this observation have in terms of understanding our risk of exposure to these tick-borne diseases? And one big answer, or potential answer to the so what question, is the possibility that there may be co-infection of ticks with more than one tick-borne pathogen. Given that these hosts are such good breeding grounds for all 3 of these tick-borne pathogens, it seems possible that larval ticks biting one of these hosts, a mouse, chipmunk, or a shrew, might acquire more than one agent, one of these pathogens, from that single blood meal, in which case it will molt into a nymph that is capable of transmitting more than one pathogen to the next unwitting victim, who could be you or me. So we've addressed the question of what is the degree to which we see co-infection or infection in ticks with more than one pathogen simultaneously in our local sites. So we've gone out for several years in a row and sampled 188 sites that are scattered throughout Dutchess County and collected large numbers of host-seeking nymphal ticks. These are the ones that are on the ground or on low vegetation looking for something to bite. And we've asked the question, what is the pattern of infection with each of these 3 tick-borne pathogens? Now what we find is, across the board, this is a very large sample of over 4,000 questing or host-seeking nymphs. We find that about 8% of them are infected with anaplasma, about 12% are infected with Babesia, and about 28% are infected with Borrelia, the agent of Lyme disease. Now, if we want to know the expected degree to which these ticks should be hosting more than one pathogen, we can borrow from basic probability theory, which tells us that the probability of two independent events is simply the product of the independent probabilities. So, for instance, if Borrelia and Babesia are being transmitted independently into ticks, then we expect the probability of a tick having both Babesia and Borrelia to be simply 12% times 28%, which is 3%. So that's our expectation. 3% of ticks should be infected with both Babesia and Borrelia if transmission of those two pathogens is occurring independently. What we find is, in fact, the proportion of ticks that are infected with both Babesia and Borrelia is about twice that, at about 6%. And in fact, looking across the board, we see that for Lyme and Anaplasma, the percentage of ticks that are jointly infected with those two pathogens is about 20% higher than expected based on independent transmission. And for Lyme and Babesia, or a triple infection with all three of these pathogens, it's nearly twice as high as one would expect based on independent transmission. And this is, and we also know where those co-infections come from. So this is, once again, this is a huge sample, over 3,000 ticks from 181 known hosts, where we ask, where are these multiply infected ticks coming from? Which hosts are making them infected with more than one pathogen? And all the action here is in the left side, this panel with the small mammals. Very few of these multiply infected ticks are coming from medium-sized mammals, larger rodents, or birds. And in fact, if you just look at the bars for white-footed mice, those are these bars right here, we see that 22% of the larval ticks that feed on a white-footed mouse get jointly infected with Babesia and Borrelia, the agent of Lyme disease. And fully 30% of the ticks that feed on white-footed mice are co-infected with some combination of two or three of these pathogens. So one of the big answers to the so what question is that the joint identity of the best reservoirs for all three of these tick-borne pathogens means that there is rampant co-infection in our ticks. And this is an important observation for both healthcare practitioners and for the public, knowing that the potential is high that a tick bite in Dutchess County might result in a patient being infected with more than one of these pathogens, means that diagnostics need to be sought in many cases for more than one pathogen, and treatment might be quite different as a consequence. So a very quick 5-second break. That was part one of this presentation. That might not even have been 5 seconds, but we'll move quickly on to part two of the ecology of tick-borne disease risk in the Hudson Valley. So what I've described to you in the first half is a pattern whereby the same group of small mammals are the best reservoirs for the agents of Lyme disease, babesiosis, and anaplasmosis. We've also asked the question in my lab whether hosts differ in their quality not only for these pathogens, the degree to which they will support pathogen populations inside their bodies, but the degree to which they'll support feeding by larval black-legged ticks outside their bodies. And this slide shows you a typical white-footed mouse from the Hudson Valley. This animal is covered with larval black-legged ticks all over its ears. And we're going to ask the question about whether different hosts support feeding by these larval black-legged ticks to a different degree. And we call this host permissiveness. To what extent are hosts permissive of larval ticks that are trying to take a blood meal off these animals? So what we do to get at this question is we go out into the field once again in August, and we capture a group, individuals belonging to 6 different species that are representative of different body sizes, different types of animals. We catch wild white-footed mice, chipmunks, veeries, gray squirrels, opossums, and catbirds. We hold them in the lab for about 3 days, which is enough time for all of the naturally occurring ticks that are on them, sometimes very many, to have dropped off the tick. And now these animals are tick-free. We then inoculate them with 100 locally collected larval ticks, and here I'm showing a veery that's being inoculated. We simply use a paintbrush to put 100 larval ticks on the nape of the neck of these hosts, and then we follow over the next several days the fate of these 100 larval ticks, and there are 2 main categories. Some of the ticks survive the experience and they feed either partially or to full repletion, and the remaining ticks die, typically as a consequence of grooming by the host. And then again, once again, we release these host animals at their point of capture. And what we find is that there's enormous variation in what we're calling permissiveness, the probability that these larval ticks will survive their attempt to feed on these hosts. So we found that 50% of the larval ticks that we placed on a white-footed mouse survived the experience and turned into an engorged larval tick, later into a nymph. That was significantly higher than was the case for any of these other hosts. Our 2 birds and chipmunks, and to some extent squirrels, were moderately permissive hosts. Opossums, on the other hand, killed more than 95% of the larval ticks that we placed on them during the course of their grooming. So we know now that these 3 hosts are the best reservoirs for the 3 tick-borne pathogens that I've described, and that mice in particular are the most permissive hosts for feeding by larval ticks. So let's address the question, so what, part 2. Why does this matter further from the issue of co-infection that I discussed earlier? Another potential consequence of this observation is that we might see increased risk of human exposure to tick-borne disease as we lose biodiversity in our landscapes. And the reasoning here is that these 3 small mammal hosts share some life history features in common, and that is they have what we consider fast life histories. What that means is that they tend to reach reproductive maturity at a very young age. Typically a few weeks of age is all it takes for these guys to mature. At that point, they start having babies. They have large litters very frequently, sometimes every few weeks to maybe a month, and they continue to crank out babies until they drop dead, possibly from exhaustion. So these animals are excellent at filling up the world with more of their own very quickly, and they tend to reach very high abundance, even though that abundance may fluctuate from year to year. And that's what we mean by a fast life history. It turns out that there's a large amount of ecological research that suggests that it is those very life history features, living fast, dying young, making lots of babies, filling up the world with more of your own, that makes animals more resilient to disturbance, degradation of the natural environment, and consequently to biodiversity loss. So to the extent that that's true, then what we should see is a dominance by these small mammal species in low diversity communities. They should be the ones that persist when we disturb the landscape or the habitat in such a way that we lose species. We should lose other species, but not them. So we've actually asked this question empirically. We've gone out into the northeastern area and asked, to what extent do we see a difference in the probability that species will disappear when we chop up the forest into smaller and smaller bits? So we selected 40 sites in the tri-state area. And those sites were forest fragments ranging in size from less than a hectare or an acre or so up to a couple of hundred hectares or several hundred acres. And we've gone out and very comprehensively assessed the diversity of mammals and ground-dwelling birds. These are the potential hosts for ticks that occur in those sites. And I'm representing here our results by a simple matrix that I'll try to describe. So what we're looking at here are, we arrange the species within the fragments where each species is a column and each row is a fragment. And we're mapping the species that occur in each of these fragments in this way. So for instance, there was one species that was found in every single fragment in which we investigated. So this is the one species that is found across the board in every single fragment. Others were found in very few fragments. A couple were found only in 1 or 2 fragments. And then this was our most diverse fragment. So just counting up the species, we find that this fragment going across the top had the most species. And it happened to be the largest fragment in our collection of 40. So we're asking the question here, as we lose diversity, so we're going from these very diverse fragments to much less diverse fragments, do we lose species in random order? Is it just all random? Or do we actually see particular species disappear and other species tend to persist? In other words, is it nested? Is there a nested subset of species as we lose diversity going from high to low diversity fragments? And we find that it is distinctly non-random, that we tend to lose particular species as we fragment the landscape, and we tend to see particular other species remaining. And in fact, what we saw in this particular data set was that 3 of the 4 most ubiquitous species that persisted even when we reduced diversity were the white-footed mouse, the eastern chipmunk, and the short-tailed shrew. So the way this looks in a sort of a cartoon version is that as we go from very large, extensive patches of forest to smaller and smaller bits of just a few hectares or maybe even less than a hectare, we see the disappearance of some species that are unable to persist and maintain viable populations in smaller fragments. We get a full complement of host species in larger fragments. But as we chop them up into smaller bits, many of these species disappear. The ones that tend to remain are these small mammals, the mice in particular, chipmunks, and to some degree the shrews as well. So the expectation then when it comes to tick-borne disease is that these small fragments should be the riskiest places. They should be the spots where we see dominance by the best reservoirs for infection and the best hosts for larval tick feeding. And in fact, this observation probably explains an observation that we made more than 10 years ago in Dutchess County, where we found that in very small forest fragments, over to the left, fragments less than about 2 or 3 hectares in size, we saw elevated abundances of infected nymphs. This is infected with the Lyme disease spirochete. But in somewhat larger forest fragments, we saw reduced densities or abundances of infected nymphal ticks. And in fact, this research, the relationship between diversity and disease risk, has been studied in other systems more recently. A group out of the University of Toronto has looked at the relationship between host diversity and Lyme disease risk in a series of islands in the St. Lawrence River that separates New York State from Quebec. And they looked at the small mammal diversity and the abundance of infected nymphal ticks across that series of islands where diversity varied naturally. And what they found was that on any islands where diversity of small mammals was low, so here we're seeing where the number of small mammal species was only about 2 or 3, In those kinds of islands, you saw very elevated numbers of infected nymphs and therefore risk of human exposure to Lyme disease. On the other hand, on islands that had 5 or 6 species of small mammals, they tended to see very low numbers of infected nymphs in the environment and therefore lower disease risk. And another even more recent study, another group from Canada, this time McGill University, looked at Lyme disease incidence, or the number of cases per 100,000 people in the eastern United States. They did this in 1992 and in 2011, looking at the CDC statistics, and asked how the incidence of Lyme disease cases in a state might be related to the number of species of hosts, host species richness as it's called, in those particular states. And what they found was a very similar pattern, but at this enormous spatial scale, where states that had relatively few host species tended to have higher incidence of Lyme disease in the human population. That pattern was found, was a dramatic one in 1992, but it became stronger through time, such that if you happen to live in a state with low species richness of hosts, then you are at much more risk of getting Lyme disease than was the case if you lived in a state that had high species richness, high diversity of these animal hosts. What this general result means is that, all else equal, preserving animal diversity, mammal and bird diversity, will tend to reduce the risk of tick-borne diseases, at least as has been documented for Lyme disease. So lastly, I want to very quickly turn to another issue that has to do with anthropogenic impacts on the environment, and this is burgeoning research on the impacts of climate change on ticks and tick-borne disease. I was privileged to be a participant in the U.S. Global Climate Change Impacts Assessment, the National Climate Assessment that was released about a year ago. And this document established for the United States very strong evidence that throughout almost all regions, with one exception being the southeastern United States, that recent decades have seen a dramatic increase in the average temperature over land in the United States. This is certainly the case in the Northeast, where we've seen growing anomalies towards more and more warmer temperatures and a steady increase in average temperature over the most recent decades. Now, this may have relevance for tick-borne disease because climate is important for the ticks in particular, potentially also for their hosts and even for the pathogens. This is one of a number of different maps that researchers have drawn in recent years of the current distribution of Lyme disease infected ticks in the United States. There are a couple of major epicenters in the upper Midwest and in the Northeast and Mid-Atlantic regions. There are various models, computer-generated models, that map the distribution of ticks currently onto current climatic conditions and then ask how these ticks should change in their distribution as the climate warms, again, as predicted by various climate models. And this just shows one such attempt. All of these different computer simulation models make similar predictions. The details differ, but in all cases, the distribution of ticks is expected to increase dramatically in the coming decades as a function of a warming climate. In particular, ticks should expand throughout the rest of New England, into western New York, up into southern Canada. Interestingly, there are a few cases that will become less hospitable to ticks, particularly some spots in the deep south, but most of this range in North America is expected to be more hospitable to ticks, giving rise to an expansion. We have asked for our local data in Dutchess County, New York, what does the warming trend tell us about the likelihood that ticks might change in their behavior, in their natural history, and in our risk of exposure. So these are data from a weather station at the Cary Institute run by Vicki Kelly, and they show that there has been a steady increase with some fluctuations in the overall temperature. These are cumulative degree days through August, and this is the overall temperature each year relative to the larval ticks. This is relevant for larval emergence, and these are cumulative degree days through May, and that's relevant to the emergence of nymphal ticks. And so we see a significant directional increase in overall temperature over the past 20 years. We've asked what impact do these data, these cumulative degree days through August or May, have on the day in which the day of the peak of either larval or nymphal host-seeking activity. And what we find in this fairly complicated slide is that as the year gets warmer, so moving from left to right along this horizontal axis, that's cumulative degree days through August or through May. As the year is warmer, we see the day of the larval peak or the day of the nymphal peak get earlier and earlier. And in fact, over the past 20 years, larvae and nymphs have both appeared about 3 weeks earlier in warmer years than in cooler years. So projecting this off into the future, what we find is that just focusing on the nymphs, these are most relevant to our health. What we find is that currently the nymphal peak is about May 24th. It's toward the end of May. Now this is about 3 weeks earlier than it used to be when the climate was a little bit cooler. Projecting out a few decades from now, we expect that the nymphal peak will be more towards the early to middle part of May, almost a full month earlier than it was a mere 20 years ago. And we think this is important for public health because if the nymphs are now emerging, they're starting to emerge in April and they're reaching a peak in early to mid-May, we probably need to think about moving Lyme Awareness Month from May into April, and that's what we have begun to recommend as a consequence of this climate warming trend. So let me quickly wrap up. What I've described is that we've seen in Dutchess County as well as elsewhere in the Northeast and on the planet, we're seeing wave after wave of emergence and spread of tick-borne disease. Of the ones that I've discussed, the 3 major ones in our region, Lyme, babesiosis, and anaplasmosis, the pathogens come predominantly from small mammals. These mammals tend to thrive in suburbanized landscapes where biodiversity is reduced. That results in an increase in risk as we degrade and fragment the landscape, and it also results in an increase in the likelihood of co-infection in ticks and possibly, therefore, in people. The models show, well, we know that Lyme disease is marching north and west with climate change, and the models suggest that this will continue on into the coming decades, so we should be prepared to be able to deal with new areas being invaded. And in the Hudson Valley, our data from the Cary Institute show that ticks are appearing earlier in the season as the climate warms, and we should probably start educating the public and healthcare providers earlier in the season as a result. So I just am flashing a picture of key collaborators and key funding sources for the research that I've described, and then at this point I will finish. And I think it is up to me to introduce the next speaker in the webinar this morning. And she is Dr. Holly Ahern, who is Associate Professor of Microbiology at SUNY Adirondack, and she will be discussing the bacteriology of borreliosis.